Assalamu alaikum warahmatullahi wabarakatuh. So today we are going to talk about collagen fibers synthesis. So before we talk about collagen fibers, we are going to revise connective tissue. So what is connective tissue? Connective tissue basically consists of four. There are four different types of connective tissue: blood, bone, cartilage, and connective tissue proper. Connective tissue is made up of cells and extracellular matrix. Extracellular matrix consists of ground substance and fibers. Ground substance is made up of interstitial fluid, cell adhesion proteins, and proteoglycans. The fibers are present or embedded within the ground substance. We have different types of fibers in connective tissue, such as elastic fibers made of elastin protein, collagen fiber made of collagen protein, and reticular fibers. The second component of connective tissue is cells. So we have two types. We have cells which can be immature and mature cells. Immature cells are called as with the ending blast, and mature cells end with the ending site. Immature cells are the cells which are very highly mitotic and they secrete the ground substance and fibers, which we have studied in extracellular matrix component. So what are the cells? So the connective tissue proper is made up of fibroblast, that is the immature form, or and the mature form is fibrocyte. Cartilage is made up of chondroblast and the mature form is chondrocyte. Bones are made up of osteoblast and the mature form is osteocyte, and blood is made up of hematopoietic cells. Now we will study about what we call types of connective tissue and their subcategories. So connective tissue proper is made up, there are two types of connective tissue proper. We have loose connective tissue and dense connective tissue. Loose connective tissue can be further divided into areolar, adipose, and reticular. Dense connective tissue proper can be regular, irregular, elastic. Then we move on to cartilage. We have three types of cartilage: hyaline cartilage, elastic cartilage, and fibrocartilage. Bones also we have of two types. We have compact bones and spongy bones. So this was a quick recap about connective tissue. So we have two connective tissue. We have connective tissue four types: blood, bone, cartilage. Connective tissue proper. Connective tissue consists of cells and extracellular matrix. Extracellular matrix consists of ground substance and fibers. And then we have the cells. So now we will coming back to our discussion on collagen synthesis. So collagen is basically triple helix alpha chain. So there are three alpha chain which forms the helix, that is collagen protein. To synthesize one type of collagen, cells have to activate different collagen genes or alpha genes. Many alpha genes for collagen are present on different chromosomes and in different cells. So therefore different cells will activate different alpha genes and they will make different types of alpha peptide or alpha protein. For example, as you can see here this alpha, this is also what we call collagen protein, this is also collagen protein, this is also collagen protein. All three are helix, that means all three have three peptide chains, but all three look different, but they are collagen, what we call protein. Here in this we have alpha 1, alpha 2, alpha 3, because all three are different. Here we have two similar chains and one different chain, and here we have one chain which is what we call two similar chains and one different chain. So it can be like this can be one of the ways of what we call types of collagen protein. So alpha 1, alpha 2, and alpha 3, what we call alpha chains, are slightly different from each other because when they were coded from the DNA sequence, their DNA sequences were slightly different, and furthermore when they formed the mRNA was slightly different, and eventually the amino acid sequence will also be different. So that's the reason why all the three chains are different from each other slightly. Collagen is the most abundant protein in our body. It accounts for almost 25% of dry weight of our body. Fibroblasts produce collagen. Other cells also produce collagen, such as endothelial cell, osteoblast, chondroblasts. And then we know that genes are sequence of nucleotide within the DNA, or gene is a specific nucleotide sequence within the DNA which is concerned with the formation of a specific RNA. So we know that collagen is basically triple helix alpha chain. So let us now look into a detailed what we call process of synthesis of collagen in fibroblasts. So endoplasmic reticulum is a network of tube inside the cell. The endoplasmic reticulum, which has ribosomes attached on its surface, is called as rough endoplasmic reticulum, and the endoplasmic reticulum where there are no ribosomes attached on its surface is called smooth endoplasmic reticulum. So here we can see smooth endoplasmic reticulum and rough endoplasmic reticulum with ribosomes attached on its surface. So now we know that if this is the fibroblast, within the fibroblast, the nucleus will contain the DNA, and when the DNA transcription occurs, we get mRNA. And then when this mRNA comes to the cytoplasm, it is going to bind with free ribosomes, which are going to translate this mRNA. So as we are translating the mRNA, we get a peptide chain, and that peptide chain at the end will have a signal sequence. This one mRNA can be attached to many ribosomes simultaneously. So, for example, if this is the first, second, and third ribosome. So, if the mRNA is moving in this direction, from right to left, so first ribosome has read most of the mRNA sequence, so it has the longest peptide. And the second one has read what we call less as compared to the first one, so it has second biggest chain of peptides, and the third one has read the mRNA chain the least, so it has the smallest peptide chain. But the thing is to remember, all of them will form a signal sequence, that is specific nucleotide sequence, which is called as signal sequence at the end of the peptide chain. And then we remember that this signal sequence is going to be identified by signal recognizing protein, and then this signal recognizing protein is going to pull this peptide chain towards the membrane of rough endoplasmic reticulum, and the rough endoplasmic reticulum has signal recognizing protein receptor, and the signal recognizing protein receptor is going to bind with the signal recognizing protein, and as a result the translocon will open, and then the ribosome and the what we call our peptide chain will enter into the rough endoplasmic reticulum. So before entering into rough endoplasmic reticulum, this peptide chain is called as pre-pro alpha peptide. It is called as pre-pro alpha peptide. But once it has entered, after translocon open, after this pre-pro alpha peptide chain enters into the lumen of rough endoplasmic reticulum, the signal sequence is removed, and it is called as pro alpha peptide. So some basic concepts that we have to remember, that one mRNA can have many what we call ribosomes attached to it. For example, if one mRNA has 10 ribosomes attached to it, so there will be synthesis of 10 peptide chains simultaneously. And also to make collagen having alpha 1, alpha 2 and alpha 3 what we call chains, three different genes within the DNA will be activated. For example, gene 1 can be responsible for synthesis of alpha 1 chain, gene 2 for alpha 2 chain, and gene 3 for alpha 3 chain. So gene 1 will make the mRNA for alpha 1 chain, that will be translated by many ribosomes, will have many peptide chains being synthesized. Same goes for gene 2, which will synthesize mRNA for alpha 2 chain, and gene 3, which will synthesize the chain for alpha 3 peptide. Now coming back to our collagen synthesis, so now we'll just consider one of the chain, for example alpha 1 chain. So mRNA of alpha 1 chain was acted upon by ribosomes. The ribosomes formed the peptide chains, and in the end they had the signal sequence. The signal sequence was recognized by the signal recognizing protein. So the signal sequence was recognized by the signal recognizing protein. This signal recognizing protein will pull the peptide chain along with the ribosome and the signal sequence towards the membrane of rough endoplasmic reticulum, and bind the signal recognizing protein to signal recognizing protein receptor, the red color one, and the black one was our signal recognizing protein. And then when they bind, they have GTP attached to them, which is released, and that gives us energy, due to which the translocon opens, and the ribosome along with the peptide chain with the signal sequence aligns above the translocon, and it enters into the lumen of the rough endoplasmic reticulum. And this peptide along with the signal sequence was called as pre-pro alpha peptide. But after we remove the signal sequence, it will be called as pro alpha peptide. So here is that pro alpha peptide within the lumen of rough endoplasmic reticulum. So now we will discuss the sequence of amino acid within this what we call alpha peptide chain or collagen peptide. So the sequence of amino acid in alpha peptide is that glycine is present at every third position. For example, if glycine is here, XY and again glycine, XY again glycine. Now why is this so? Why do we have glycine present at every third position within the alpha peptide chain? This is because glycine is a small glycine is an amino acid which has a small side chain. So when we twist these three chains tightly, Because these glycine have a short side chain, they can even what we call lock within themselves. So that will provide further strength or tight attachment. This is why glycine is present at every third position. So that this is the reason why glycine is present, because if we would have taken some other amino acid and that would have a longer side chain, it would not twist that strongly. For example, if three fat people hug versus three thin people, we know that three thin people will hug each other more tightly as compared to fat people. Same goes that a bigger amino acid will have a longer side chain, so they can't twist together as well as the glycine. Now it is so important that if this presence of glycine is what we call nucleotide is mutated, we will have some disease also. For example, if the DNA sequence of this what we call alpha peptide, which will code for this alpha peptide, the DNA sequence which makes the mRNA was mutated and there was no glycine, eventually we will have a disease, because the collagen fiber which will be formed will not be very tight and it will be very not be as strong when there is glycine present, because glycine helps in the stronger tightening or stronger tight attachment of the collagen. So if there is disease in which the nucleotide coding for glycine is muted and instead of glycine at every third position we can have some other amino acid, the collagen formed will be weak due to weak attachment with each other of the collagen, due to weak attachment of the alpha chains with each other, the collagen will be weak, and if the collagen is weak, the bones will be weak, and as a result some fetus can undergo fracture which can occur after birth or even before birth. This disease is called as osteogenesis imperfecta. So osteogenesis imperfecta is a bone formation disease in which bone formation is imperfect due to mutation in genes which code for glycine amino acid of alpha chains, especially of the collagen which is present in bone. In bones, the collagen which is present is type 1 collagen, because bone itself in the spelling you can BONE. So common sense, that's a helpful point you can remember, as type 1 collagen is present in bone. So such people whose type 1 collagen which is present in bones is mutated or has some problem, the triple helix will be not be very tight and they will have osteogenesis imperfecta. Now the degree of mutation can be varied. It can be mild, moderate and severe. And osteogenesis imperfecta is of two types. We have osteogenesis imperfecta congenita, that is the baby dies within the uterus itself because he has such weak bones that his skull crushes and the brain gets damaged or the ribs break within the within the uterus, so it's intrauterus death, and it is called as osteogenesis imperfecta congenita. Then we have osteogenesis imperfecta tarda, that this happens after the birth, that the baby survives till birth but his bones are still weak. So example, if you hug the baby, maybe his ribs will break, or if the baby falls down, the sternum breaks. And usually when such patients are taken to hospital, the doctors usually confuse with the diagnosis and they think that the baby has undergone child abuse, and there can be made a case against it. So that is osteogenesis imperfecta tarda. So as we discussed about the pro alpha peptide, and in alpha peptide we discussed that what we call on its amino acid sequence of the alpha peptide, we remember that every third position is glycine, and we have to remember that the Y position we can have either proline or lysine. So at the Y position we can have either proline or lysine. Now we come to the second important what we call process of formation of the collagen, that is hydroxylation. So hydroxylation is basically attachment of hydroxyl group to either proline or lysine which is present at the Y position, and these hooks are very important for further tight attachment. So it is very important for strengthening of the collagen molecule. So we remember that the pro alpha peptide chain had glycine at every third position, and that was because it has a small side chain to help in further when three peptide chain, three helixes form, so they will be further tightened. And now we further attach hydroxyl groups and do hydroxylation to further strengthen that twisting of the three alpha peptide chains when they attach with each other. So here we can see hydroxylation is being done, and this hydroxylation will be done at the Y position as I mentioned, and it will be done by either lysyl hydroxylase for lysine and or prolyl hydroxylase for proline. But hydroxylation of pro-alpha peptide by hydroxylases such as prolyl hydroxylase or what we call lysyl hydroxylase requires a cofactor, and that cofactor is vitamin C. So you can see here vitamin C, it is a cofactor. Vitamin C or ascorbic acid. So basically cofactor means that if vitamin C is not present, lysyl hydroxylases and prolyl hydroxylase will not hydroxylize the pro alpha peptide chain. So this is a pro alpha peptide chain, and here in blue we saw that they hydroxylated them, and then they are acting as hooks, those hydroxyl groups. But if vitamin C is not present, lysyl hydroxylase and prolyl hydroxylases will not hydroxylize the pro alpha peptide chain. But this does not mean that collagen is not being produced. Collagen will be produced, but it will not be very strong, and it won't be very, what we call, its strength will be decreased because it won't be hydroxylated and it won't have these extra hooks. So as a result, the collagen being produced will be loose if there is no hydroxylation done. So due to deficiency of vitamin C, we can suffer with scurvy. So scurvy will have clinical problems because of severe vitamin C deficiency, and this can lead to problems such as we can have, when there is vitamin C deficiency, we can have gums injured, that is tooth may start to fall due to severe deficiency of vitamin C. There can be microhemorrhage because we know that capillaries are what we call endothelial tubes which have collagen around them. So if collagen will not be strong, it will be loose, there will be ruptures, micro small ruptures, and endothelial, what we call capillaries will be very fragile, and as a result we will have microhemorrhage. Then we can have impaired wound healing because we know fibroblast goes to the place where there is a wound, and if fibroblast cannot produce proper collagen, again we will have poor wound healing. And sometimes severe vitamin C can also lead to death of the person. So this as a whole, the disease scurvy can have gums injured, microhemorrhage, poor wound healing, and also can lead to death. And all this was due to what we call our no hydroxylation of the pro alpha peptide chain by propyl hydroxylase and lysyl hydroxylase. Now, after prolyl hydroxylase and lysyl hydroxylase have hydroxylated the pro-alpha peptide chain. So here we can see the hydroxyl group as hooks. The next step is called as glycosylation, that is adding sugar to this pro-alpha peptide chain. So glycosylation is the addition of either glucose or galactose sugar, which are monosaccharides, and it is done by glucose transferase or galactose transferase. Now where is this glucose and galactose added? This glucose and galactose is attached to hydroxylysine. So remember we told that in the amino acid sequence of pro alpha peptide we have GXY. Here that GXY and G is glycine always, and Y can either be proline or lysine. So if the Y has lysine, and that lysine when we hydroxylate it and add OH on it will be called as hydroxylysine. So that is the place where we are going to add either glucose or galactose, and this is called as glycosylation. So here also we can see hydroxylation, and after hydroxylation we had this hooks on the pro alpha peptide chain, and now we will add sugar over there with the help of glucose transferase or galactose transferase here. Now the pro alpha peptide is what we call hydroxylated as well as glycosylated. So what happens, as I told before, multiple peptide chains were formed since multiple ribosomes were attached to a single mRNA, and those multiple chains, all of them must have entered the lumen of the rough endoplasmic reticulum. So all of them must have been hydroxylated and glycosylated. So out of the multiple alpha chains, three, because we're going to form a helix, right? So we will imagine the three of these chains, alpha chains, start to come close to each other. So here we can see these three alpha chains came close to each other, and then we all know we have a carboxyl end and an amino end. The twisting of these three chains starts from the amino end because of formation of disulfide bonds. We have disulfide bonds which will be formed on the carboxyl end as well as on amino end, but the amount of disulfide bonds will be higher on the carboxyl end, so therefore the twisting begins from the carboxyl end. And the twisting will look something like this, that the ends are free or they are loose, but the main twisting happens in the middle, and this will be called now as pro-collagen. So here we can see that we had our three peptide chains, alpha pro alpha peptide chains. After they had been hydroxyl and glycosylated coming close, and the carboxyl end had some disulfide bond and also the amino end, but they were more on the carboxyl end. They started twisting, and eventually we have this pro collagen being formed. Now this pro-collagen will bud out of the rough endoplasmic reticulum and form a transfer vesicle, and it will go to the Golgi body. And in the Golgi body, it will again be sugared, or more sugars will be added to it. And here in Golgi body we will add oligosaccharide, that is many sugars are added, whereas here we added monosaccharide. We just added in glycosylation, we had added glucose and galactose, which are monosaccharides, but here we will add long chains of sugar, that is more oligosaccharides, to this pro-collagen. And then that pro-collagen will form a secretory vesicle, and they will be eventually secreted out of the cell of the fibroblast. This secretory vesicle has a special transport mechanism called as microtubules or microfilament, which help it to go out of the cell rather than going back into the cytoplasm of the cell. So with the help of these microtubules or microfilament, the secretory vesicle will reach the cell surface or cell membrane, and both of them, the secretory vesicle and cell membrane, will fuse with each other, and the pro-collagen will be released out of the fibroblast cell. The thing to remember is that microfilaments are actin, made up of actin mostly, and microtubules are made up of tubulin mostly. So here we had either microfilaments or microtubules, and then our secretory vesicle fused with the cell membrane and procollagen was released. Now if we will go fast for a recap, we had our nucleus of fibroblast, in which the DNA underwent transcription to produce mRNA of alpha chain, and that mRNA was translated by many ribosomes, and it was producing multiple peptide chain. Each peptide chain has signal sequence at the end, and this signal sequence was recognized by signal recognizing protein, and that signal recognizing protein pulled this full complex of the signal sequence along with the peptide chain and ribosome towards the rough endoplasmic reticulum membrane, which had the sequence recognizing protein receptor, and when both of them bind, they released energy to open a translocon, and eventually our pre-pro alpha peptide chain entered into the lumen of the rough endoplasmic reticulum. And once it enters the lumen of endoplasmic reticulum, signal sequence is cut, and this peptide chain is now called as pro alpha peptide chain. And then pro alpha peptide chain will first undergo hydroxylation at its Y position, which has proline, or lysine, which is done by lysyl hydroxylases or prolyl hydroxylases, and vitamin C acts as a cofactor for lysyl hydroxylases and prolyl hydroxylases, and in the absence of vitamin C, prolyl hydroxylases and lysyl hydroxylases will not hydroxylate propeptide. After that, the pro-alpha peptide underwent glycosylation, where glucose and galactose monosaccharides were added on hydroxylysine, and then multiple chains since came into the lumen of rough endoplasmic reticulum. They came close to each other and formed disulfide bonds, mostly at the carboxyl end and some at amino end, and from there they started twisting into a triple helix, and pro-collagen was formed. Then pro-collagen was released from the rough endoplasmic reticulum in a transfer vesicle, and this transfer vesicle went to the Golgi body, and in Golgi body it had multiple or many monosaccharides called as oligosaccharides attached to this pro-collagen, and then pro-collagen went out of the cell via secretory vesicle on special transport mechanism called as microtubules or microfilament. So this was the intracellular part of collagen synthesis. Now we are going to see the extracellular part of collagen synthesis. So once the procollagen was secreted by the secretory vesicles and it comes out on the surface of the cell, there are enzymes called collagen peptidases, and these collagen peptidases will cut the free or the loose part of the procollagen. And after we cut the loose part, we will have only the vertical triple helix part, which is very tight, and that is called as tropocollagen. And the free or the loose part of the procollagen which was cut is called as propeptides. Now what happens is many of the tropocollagens, from now on we will represent like this, many tropocollagens start to come close by, and they are what we call fixed together tightly with the help of the hydroxylation bond which we had formed during the intracellular part of collagen synthesis. And not only this, there is an enzyme called a lysyl oxidase, and what this lysyl oxidase does is, lysyl oxidase is a copper dependent enzyme, and it works on lysine and hydroxylysine of tropocollagen and converts them to aldehyde. As a result, many tropoglycans will form strong covalent bonds among the aldehydes which have been formed by lysyl oxidase. So lysyl oxidase will convert the lysine and hydroxylysine into aldehydes, and aldehydes of respective tropoglycans, tropocollagen will form strong covalent bonds among themselves. And also this is the extracellular mechanism of strengthening of the what we call tropocollagen. The intracellular mechanism of strengthening, we had the hydroxylation in which had given small hooks to proline and lysine, and which was catalyzed by vitamin C. Extracellularly, lysyl oxidase converts the lysine and hydroxylysine to aldehyde, and these aldehydes of different tropoglycans will form strong covalent bond, and which gives ultimate strength to the collagen. And then when we arrange many tropocollagen together, it is called as collagen fibril, and many collagen fibrils, if we arrange them, we will get the collagen fiber. So now we know that the DNA, what we call transcription, led to the formation of our mRNA, and that mRNA, after it came to the cytoplasm, it was acted upon by ribosomes and translated in single sequence, and that when it bind to the rough endoplasmic reticulum, and this peptide sequence was called as pre-pro alpha peptide. But then once the pre-pro alpha peptide comes into the lumen of rough endoplasmic reticulum, single sequence is gone, it is called as pro alpha peptide. This pro alpha peptide will be undergone hydroxylation by lysyl hydroxylases and prolyl hydroxylases on proline and lysine, and that is done with help of vitamin C, which acts as a cofactor. And after hydroxylation, we have glycosylation, which is addition of monosaccharides on what we call hydroxylysines, and then many multiple chains or three chains will come together and they will undergo what we call twisting, and that starts from the carboxyl end due to more sulfide bonds present at that end. And then we will have procollagen, which will have central twisted part and ends will be loose or free, and then the rough endoplasmic reticulum will form a transfer vesicle, and the transfer vesicle will contain the procollagen, which will go to the Golgi body. Golgi body will lead to the addition of oligosaccharides in the procollagen, and then the procollagen will be secreted via secretory vesicles, which will be transported with the help of microtubules of filament, and once it comes out, it is procollagen, and the collagen peptidase will act on procollagen and separate the propeptides and form tropocollagen. Then many tropocollagens will come together, and they are what we call strengthened due to hydroxylation of the hydroxylysines and hydroxy prolines. But meanwhile, lysyl oxidase will convert lysines and hydroxylysines into aldehydes, and aldehydes of what we call adjacent tropocollagen will form strong bonds, and many tropoglycans tropocollagen together form collagen fibril, and many collagen fibril together form the collagen fiber. Now we can discuss some diseases associated with extracellular mechanism of collagen synthesis. So if there is a problem with the enzyme collagen peptidase or lysyl oxidase, that either their DNA, the sequence had been mutated, or some other problem, or they are dysfunctional, then the collagen which will be formed till here, procollagen, that will be normal, but after that the further processes will be disrupted and we will have eventually weak collagen. So if either of these enzymes, either collagen peptidase or lysyl oxidase have a problem, we can have the disease Ehlers-Danlos syndrome. So in Ehlers-Danlos syndrome, the problem is basically that after the pro-collagen is formed, there is problem with the processing of the collagen, and eventually the collagen will be weak, and this happens mostly for collagen type 3. And due to this, the patient will have stretchable skin, the patient will have mobile joint which are hypermobile, and there is a high risk of rupture of internal organs of such patient. There is pathological stretchable skin of the patient, and the bending of joints of the patient is also hypermobile mobility. That is, you can even bend the joints in a forward way more than their limit. And this is because bones are made of collagen, capsule is made of collagen, ligament and tendons are also made of collagen, and all are weak, so joint becomes more hyperextensive and hypermobile. Not only this, collagen type 3 is used mostly for supporting the viscera like gastrointestinal tract, vascular system, etc. So if collagen type 3 is weak, then what we call, there can be high risk of rupture of GIT, aorta, aortic vessels, etc. But if we compare this Ehlers-Danlos syndrome with osteogenesis imperfecta is more dangerous because their bone or collagen are even more weaker as compared to Ehlers-Danlos syndrome, because there the gene for glycine was mutated. So during the intracellular synthesis of collagen stage only, there will be poor what we call twisting of the collagen fibers among themselves, and as a result the pro-collagen will also be very weak. Here in Ehlers-Danlos, the till pro-collagen the collagen was normal, it was perfect, but after that there can either be a problem in collagen peptidase or lysyl oxidase that affects the further processing of collagen. Osteogenesis imperfecta is called as brittle bone syndrome also, and there was the tendency of undergoing multiple fracture. And also in osteogenesis imperfecta, the patient's sclera will be blue, not his lens or eyes. His sclera in his eyes is going to be blue, and that is because collagen type 1 is affected in osteogenesis imperfecta, and collagen which is present in the sclera, which makes the white part of the eye, it will be very low and not perfect, and so the veins underneath the sclera will be able will be seen, and as a result the venous blood will be reflected and we feel like the sclera is blue, and that happens in osteogenesis imperfecta. And another type of what we call collagenopathy is scurvy. We remember scurvy. Collagenopathies are basically problems with collagen that can be viscerally osteogenesis imperfecta. We discussed Ehlers-Danlos syndrome, and we had discussed scurvy also. So scurvy was basically that due to deficiency of vitamin C, lysyl hydroxylase and prolyl hydroxylase will not hydroxylate the pro alpha peptide chain, and as a result we won't have what we call these small hooks that will form strong pro collagen. So these were some diseases associated with collagen problems. Now we will recap the mechanisms of strengthening the collagen fiber. So the first mechanism was hydroxylation, and then after that what happens is, what I forgot to mention was that pro alpha peptide chains, the single chains are all left handed twisted, okay, they're twisted in the left hand direction. But when we take three pro alpha peptide chains, okay, and we are going to form a helix, all of them singularly will be left handed twisted, but together they'll be twisted in the right hand direction, so that gives extra strength. And such mechanism we see in modern day infrastructure such as in bridges. For the suspension bridges, the steel rods are done like this. So this is a very important way of adding strength to the collagen fiber. So once again, coming back to recap of how can we strengthen the collagen fiber. The first method was hydroxylation, the second was left-handed twisting of pro-alpha peptide, the third was glycosylation, and the fourth was right-handed twisting of the pro-collagen, and the fifth mechanism was what we call lysyl oxidase helps in formation of interlink between the tropocollagen by forming aldehydes with strong bonds among the tropocollagen. So these were the five mechanisms of strengthening the collagen fiber. There are many different types of collagen, around 27. Out of those, we will discuss some. So, histologically, if we see type 1, type 2, type 3, and type 4 collagen, they have fibrillar pattern. This means they have light zones and dark zones. Light zones and dark zones when they are stained and seen under microscope. And it was observed by scientists that there is a gap of 67 nanometer, and after every 67 nanometer, we'll have a dark zone, and again, then 67 nanometer light zone, again, dark zone. So this is the called as fibrillar arrangement or fibrillar collagen pattern, which is seen in type 1, type 2, type 3, and type 5 collagen. Type 4 and type 7 collagen form meshwork or network type of pattern, and it is present in few places such as in the glomeruli of the glomerular basement. In kidneys it has type 4 of collagen because that helps in passage of materials, selective materials through the glomerular basement membrane. And then it is also seen in capillaries. So around the capillaries we have type 4 connective tissue or type 4 collagen fiber, I'm sorry, because it is a mesh work, right? It's like a net, so it helps in movement of materials from the capillaries to the tissue and from the tissue to the capillaries. That is why we use type 4 collagen fibers in kidney glomerular basement membrane and in cells which are supported of the and in the cells around the capillaries. So all these intracellular mechanism and extracellular mechanism which we had seen, this will basically apply for type 1, type 2, type 3 and type 4 collagen fibers. Type 4 and type 7 collagen fibers are slightly different, such as in the way that intracellular mechanism collagen synthesis same, but when they come out, the pro collagen, as we remember in type 1, type 2, type 3 and type 5, they will the collagen peptides will act and cut the loose ends, that is the propeptides. But in type 4 and type 7 collagen, it doesn't happen. And in type 4 and type 7 collagen, the pro collagen will come out and it will form a dimer, that is the loose ends won't be cut and the loose ends will join with each other. And also it doesn't form a fibrillar pattern. There will be no such pattern because it will form a mesh or network. And since it doesn't form a fibrillar pattern, there will be no periodicity, that is after 67 nanometer light order, it won't be there in type 4 and type 7 collagen. So this is the basic difference, that the loose ends are not removed and they will form dimer after being released, and eventually they will form a meshwork. So there are 27 types of collagen. Out of that, we are going to discuss around 6 of them. That is 1, 2, 3, 4, 5 and 7 collagen. So the thing we have to remember is that question like is how all collagen are different from each other. This means that different collagen have different alpha peptide or combination of them. So I will explain that. For example, here we have example, we'll consider this as collagen type 1. So in collagen 1 all three are alpha 1 chains, but in collagen 2 we have two alpha 1 chains and one alpha 2 chain, and in collagen 3 we have two alpha 2 chains and one alpha 1 chain. So in such example we can understand that all collagens are triple helix of alpha peptide, but different collagen may have different combination of different alpha peptides. So we have three alpha peptides, right? We have alpha 1, alpha 2 and alpha 3. So we can arrange these three alpha peptide chains in different combination to form a different type of collagen. So now collagen number 1, as we already had discussed, it's present mostly in bones and also in skin, some not fully, but also it's present in skin. Collagen type 2 is what we call present in cartilage, and collagen type 1 is hard, and so it can withhold strong pressure. And if we have a problem of collagen 1, we will have osteogenesis imperfecta disease, like we had discussed, due to mutation of glycine gene. And collagen type 2 is associated with cartilage, which can withhold intermittent pressure. Collagen type 3 is like what we call present in viscera. It is free, it is present in many places, and it is stretchable. Collagen 3 makes the support system of GIT, it makes the support system of internal vessels. Collagen 3 also makes the support system of lymph vessels. So collagen 3 is very free and it is present around the viscera. And collagen 3 is also called as reticular fibers. And they help those organs which are like stretchable, which can stretch a bit. And also we should remember that type 3 collagen is the collagen that is laid first at the point of wound. So this means that when first we have an injury or a wound, the fibroblasts will first lay down collagen type 3, and then eventually they will switch to formation of collagen type 1, and that is due to wound maturation. So fibroblast will switch its genes from type 3 collagen to type 1 collagen genes. But sometimes it happens that in some patient there is a problem and there is excessive secretion of collagen type 3 and they will never form collagen type 1. So they form a wound which is very bad in appearance and it is an elevated scar, and that is called as keloid. So keloid is an excessive scar formation of collagen type 3, because normally we know that we had to secrete collagen type 3, and eventually that collagen type 3 fibroblasts should stop producing and they should start producing type 1 collagen, which is strong and it will shrink. But in pathological condition, in some patients there will be just type 3 collagen which will be secreted, and that will lead to the formation of a bad appearing or elevated scar called as keloid. Here we can see an example of keloid. Also here we can see another example of keloid due to excessive secretion of collagen type 3. Type 4 collagen, as we remember, forms the meshwork or network, and it is present in kidney glomerular basement membrane, or it is present in blood vessels around the capillaries, and also it is present in inner ear. Now it sometimes happens that there can be a mutation in genes which code for collagen 4 or collagen type 4. Then what will happen is that our glomerular basement membrane and inner ears and blood capillaries will not be healthy, and the patient will develop renal failure and deafness, and this is known as Alport syndrome. In this, the patients, when his glomerular basement starts damaging too much, there will be blood and hematuria and proteinuria, and eventually the patient will undergo death if not treated. Type 5 collagen is expressed in embryo. Mesenchymal cells in embryos secrete type 5 collagen. Type 7 collagen forms again a network of meshwork, and it is present between epidermis and dermis layer in the skin, and this epidermis and dermis layer are held together by collagen number 7. So if we have any genetical what we call problem or mutation in type 7 collagen, epidermal layer and dermal layer of skin won't be held together in a strong way. As a result, if there is a slight scratch or something, our epidermal layer will come off and separate from the dermis layer, and this is called as epidermolysis bullosa. Here is an example of epidermolysis bullosa where the epidermal layer has separated from the dermis.